Hello, everyone, and welcome to today's keynote webinar titled Reshaping the Immunotherapy Landscape Through Molecular Engineering. This webinar is part of the 7th Bioprocessing Virtual Event Series. I'm Matt Thorne from LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before I begin, I would like to remind everyone that this event is interactive. We do encourage you to participate by communicating with other attendees using the live chat feature during the presentation. You can find the live chat located in the bottom left of your screen. And you can also submit your questions during the presentation. To do so, you can type them into the Ask a Question box on the bottom right and click Submit. We'll try to answer as many questions as we have time for at the end of the presentation. If you're having any trouble hearing or seeing the presentation, you can click on the Help Desk button from the lobby within the event itself. As a reminder, this presentation is educational and offers free continuing education credits. You can click on the continuing education credits link located within the abstract on the left of your screen and follow those processes to obtain your credits. So now let's get started. I'd like to present today's speaker, Jamie Spangler, Associate Professor at Johns Hopkins University. For a complete biography on our speaker, you can find that also on the left side of your screen. Jamie, you can now begin your presentation.
Thank you so much, Matt, for the introduction, and thanks for this opportunity to speak to all of you guys today. As Matt said, we want this to be interactive, so please keep the questions coming, and really excited to share some of the research going on in our lab in the drug development, drug design, and therapeutic translational landscape, particularly related to proteins and the possibility of using proteins as drugs. So the opportunity, to use a protein as a drug actually did not exist until the 1980s. So prior to that, all drugs were based on small molecules or other entities, but nothing in the what's called biologics category or protein-based drugs was actually possible. And actually a lot of biomanufacturing, bioprocessing, sort of workflows were instrumental in sort of making that an actual thing. The market has been growing since the first approval of a protein-based drug was recombinant insulin in 1982. And you can see that steadily increased up until where we are today, which is that about 40% of the new molecular entities or new drugs that are approved each year are actually biologics or protein-based. And that's, a really growing number. And overall, biologics now, even though small molecules had a significant head start and still make up the majority of new molecular entities, now make up about 20% of the overall global pharmaceutical market. But they really pack a punch. If you look at the top 10 best-selling drugs on the market today, many of them for cancer immunotherapy, for arthritis, or other sort of immune inflammatory conditions, the 7 out of those 10 are actually protein-based. And those include, antibody drugs, receptor decoy drugs, other engineered or natural versions of proteins that are in our body. But proteins, right, as they exist in nature, they don't care about your drug. And in fact, there are many endogenous properties of proteins, just intrinsic to them, that actually make them terrible drugs. Oops, sorry, I think this is so annoying. Sorry, I'm going to silence my cell phone. I didn't realize, but my office phone often gives me fun spam. So that's what that was. But regardless, so proteins in nature, again, right, have no business and no pressure selectively making them into good drugs. And they actually have a bunch of properties that make them really bad drugs. So the first property is what I'm referring to as pleiotropy. And so pleiotropy can basically means that proteins can do a lot of different things in a lot of different contexts. And that's really great if you're a protein in nature and you're trying to have many different functions within a small kind of genetic package. But that's actually terrible for two reasons when you try to make a drug out of it. The first is that you're going to have very low efficacy for your given function because you're going to all of these different places, and it's preventing you from getting where you need to go in terms of the disease, for example, a tumor in the case of cancer. And also, by going to all of these different places, you're going to have terrible off-target effects, right, on all these different regions of the body. In addition, proteins have lots of issues with expression and stability, which this crowd can appreciate very strongly, which is that, the scale-up manufacturing, developability of these proteins can be very limiting, which can lead to difficulties in manufacturing, immunogenicity, and other downstream problems when you're trying to make a drug. Finally, proteins actually are very live hard, die young, right? Sort of do your function and then get cleared out of the bloodstream very rapidly and get downregulated so everything can stay in balance. And again, that's great if you're a protein in nature, but it's terrible if you want to have a long, durable response because anything under a 50 kilodalton cutoff, which encompasses most growth factors, cytokines, chemokines, it's going to disappear from the bloodstream in 5 minutes through renal clearance. So again, there's a lot of great properties of proteins that make them really good at what they do in nature that are unfavorable as drugs. And so what do we do? And the way that I sort of view this problem is, okay, nature has presented us with these proteins, and now we can either redesign the ones that nature has given us to make them better, or inspired by nature, we can actually build brand new proteins, right, that nature's never seen before that could be useful as drugs. And how do we do this? So our lab takes on sort of this three-pronged approach. The first prong is what I'm referring to as molecular engineering, which can mean a lot of different things to a lot of different people. We do a lot of molecular evolution, meaning that we take a protein shown in red, we make a whole library of different proteins shown in green and orange and cyan based on that original protein. And then we do a selection using typically the yeast surface display platform, but other display platforms such as phage or ribosome or mammalian cells is also possible. And what you do is you select out the clones that bind to your target protein shown in gray, and you wash away the ones that don't. So through these iterative cycles of essentially Darwin in a test tube, you can evolve the protein to have the particular function that you would want. And of course, we're not the first people to look into or to use display platforms to engineer proteins. But what we do, in our lab is we really try to integrate that as much as possible with structural knowledge. And that structural knowledge can come from experimental methods like crystallography, cryo-EM, NMR, or it can come from computational methods. For example, through AlphaFolds or Rosetta or other sort of protein structure prediction programs, also through molecular dynamics simulations. Again, trying to understand protein-protein interactions, either statically or dynamically, in order to make better judgments about what proteins should go into this library that we're selecting against. And ultimately, when we select those proteins from the library, modeling or solving their crystal structures to understand how they're working mechanistically and how we can make them better. And ultimately, right, we want this to go somewhere, right? So why are we doing all of this? And really at Johns Hopkins, being at a very strong medical institution, it's very embedded and ingrained in what we do, that we want to go from the discovery phase through the design and implementation, right, making the proteins actually happen, to actually testing them in mouse models, other large animal models, even using our own human peripheral blood from ourselves or from other patients, so that we can test how it would do in theory, and then ultimately to translate that into a human clinical trial and then loop back, right? Say, how did we do as a drug, and how can we iterate the cycle again to make this a better drug? And overall, our lab consists of 3 general thrusts, right, in terms of this structure-based molecular engineering for therapy. And the first arm or thrust is actually involving the immune system, and particularly soluble ligands of the immune system, such as growth factors and cytokines. And what we're trying to do In certain diseases like cancer or infectious diseases, we're trying to activate the immune system, right? Have pro-inflammatory effects to go and kill that disease and destroy that pathogen. On the flip side, we actually have many diseases where our immune system is on overdrive and actually attacking our own healthy organs. And so in those contexts, we actually want to increase the suppressive arms of the immune system, for example, through regulatory T cells or other suppressive cells that would ultimately reduce and sort of dampen an overactive immune response. The second arm actually involves antibodies and antibody design. As a manufacturable drug and molecule, antibodies really have cornered the market in terms of biologics, and for good reason. They are a very stable and pharmacologically favorable format. And they're also very modular, right? You can attach things to all the different ends of the antibodies and create bispecifics, create multifunctional antibodies that can act through very specific mechanisms simultaneously. And then finally, the third arm of the lab is actually focused on platform development. So the first two arms are more on kind of things like, can we actually make actual drugs? And then the third is, can we develop frameworks, workflows, platforms for building and discovering and designing new drugs? And that's where we've come up with several different platforms, one of which I'll talk about today, that are centered around ways that we ourselves in our lab can make protein drugs, but also that we can inspire and share these tools with other labs in both academia and industry who would be able to use these as potential frameworks for discovering their own drugs or for purposes we never could have even imagined or envisioned in our own lab. And so that's been a really, really rewarding sort of workflow. Okay, so without further ado, I want to jump into the three projects today. I'm going to go really quickly through them because we don't have tons of time, but these are all published and the publications are listed on the respective pages. And I encourage you all, if you're curious, to look into them and to email me and follow up if you have any questions. So the first story I want to talk about is our work on engineering these cytokine antibody fusion proteins to modulate the immune response, either to amplify pro-inflammatory effects or to induce immunosuppressive effects. And so in particular, we're very interested in engineering cytokines, as I mentioned. And cytokines, the way that they act is by interacting with surface-bound receptors. And that interaction with the surface-bound receptors leads to the intracellular domains of those receptors as they crash into each other on the surface, coming into contact with each other and eliciting a cascade of downstream events that ultimately converges on nuclear transcription and tells a cell what to do, right? Whether it's going to grow, differentiate, migrate, die, right? All of that is actually dictated by these structural interactions between the cytokine and its cognate cell surface receptors and the information that it transmits into the cell. A particular cytokine that's going to be the focus of the first part of this talk is what's known as interleukin-2, or IL-2 for short. IL-2 was actually the first cytokine to be structurally and molecularly and genetically characterized, and for good reason. It plays a pivotal role in the differentiation, activation, and survival of a variety of different immune cell subsets, in particular T cells and natural killer cells. And what's interesting about IL-2 is its receptor actually comes in two formats. Cells that are low in the IL-2 or alpha receptor signal through the heterodimeric receptor, consisting of just beta gamma, the minimal signaling receptor. And cells that are high in IL-2 or alpha can actually signal through this heterotrimeric complex. And what's important is there's a 10 picomolar affinity for the heterotrimeric complex, whereas there's only a 1 nanomolar affinity for the heterodimeric. So in general, endogenous IL-2 will preferentially engage with cells that have high levels of IL-2 or alpha. And this is important because whereas effector cells, things like CD4, CD8 positive T cells, as well as NK cells, actually at resting levels have very low levels of IL-2 or alpha. Regulatory T cells, or Tregs, have constitutively high levels of IL-2 or alpha. So generally, Tregs are more sensitive than effector cells to circulating IL-2. But regardless, IL-2, again, can act on both sides of the street. And as a result, it's been used both in cancer therapy and for treatment of infectious disease on the clinical levels. And conversely, it's also been used as an autoimmune disease therapy or to promote transplantation tolerance. And as you can see, right, the fact that the same molecule is being used in two different contexts or frameworks, it's very limiting, right? Because the same molecule that is activating the immune response is actively suppressing the immune response. And the same molecule that you're using to suppress the immune response can also activate those same pathogenic cells that are ultimately mediating the disease. And furthermore, IL-2 is very toxic when delivered at high levels. It induces what's called vascular leak syndrome, which is where your vasculature, as the name would suggest, becomes very leaky, leaking fluid in the interstitial space, which can ultimately cause multiple organ failure and even death. So actually, IL-2 has been an approved drug since the early 1990s. But is really not used very much at all clinically, other than as a last resort because of the devastating side effects associated with that cytokine. Okay, so I've stated the problem again. How do we address this? And how do we find a version of IL-2 or make a version of IL-2 that would be biased? Well, it turns out we didn't have to look any further than was a Western blotting antibody called S4B6 that was found to bias IL-2 towards activation of effector cells. And conversely, there was a second antibody that was found to bias IL-2 towards activating regulatory T cells. But of course, that begged the question of why is this happening? So actually, while I was a postdoctoral fellow in Chris Garcia's lab at Stanford, I had the wonderful opportunity to actually solve the crystal structure of the S4B6 antibody and the JUS6-1 antibody, shown here in terms of their variable regions, bound to IL-2. And here it's shown overlaid with the alpha beta gamma receptors. You can see that S4B6 is binding sort of on the apical end here, interfering with the IL-2 or alpha receptor, but not beta gamma. And conversely, JUS6-1 is actually binding sort of on the dorsal end here, interfering with beta gamma, but not necessarily with alpha. And so based off of sort of our structural efforts, we were able to determine the mechanism through which our molecule, not sure, was able to bias towards either an effector or a regulatory response. And in the effector case, the S4B6 actually blocks the IL-2 or alpha subunit, which is important for regulatory T cells, but not effector cells. And it also enhances the IL-2 interaction with the beta receptor. So it actually potentiates the interaction of IL-2 with effector cells. It can still interact with regulatory T cells, but the S4B6 actually blocks the epitope where IL-2 would normally engage, IL-2 or alpha, which allows for interaction with these cells, but without that benefit, right? So the Tregs have sort of lost their edge. And because the effector cells are overall more abundant and express more beta with alpha out of the question, they actually get the overall advantage in terms of immune homeostasis. Conversely, the JUS6-1 antibody, the way that that works is through this allosteric exchange mechanism, where essentially the IL-2 JUS6-1 complex cannot interact at all with an IL-2 or alpha low effector cell because the JUS6-1 is blocking the epitope where that would normally interact with the beta gamma subunits. However, the IL-2 or alpha high regulatory T cells can actually grab the IL-2, displacing the antibody. The antibody then diffuses away. This allows for a high affinity interaction between the IL-2 or alpha beta gamma chains with the IL-2, forming the high affinity heterotrimeric complex, which in turn leads to an upregulation of IL-2 or alpha on those regulatory T cells and allows for exquisitely high and exclusive signaling on Tregs and no activity on effector cells. Okay, so that's really great, but things fall apart, right? So just because we have IL-2 in complex with an antibody, it's not necessarily going to stay that way, especially when we put it in vivo. And when it falls apart, there's two problems. The first is that the complex dissociation will lead to the same IL-2, right? Toxic IL-2 that was the problem before and unbiased. And the second is that it will actually, the IL-2 half-life is about 5 minutes because it's a very small protein. And the antibody actually extends that to on the order of weeks to sometimes even months. And so again, if it dissociates, then it's going to lose that edge. And so if you can't beat them, join them, as I always say. And so that's exactly what we did. We put a nice long linker between the C terminus of IL-2, the N terminus of the light chain of either the SRB6 or JAK6 antibody to make an intramolecular immunocytokine, where the cytokine sort of folding upon itself. And this is not your typical immunocytokine, right, where the antibody is targeting some antigen and the cytokines on the C terminus along for the ride, but rather an N-terminal fusion so that the cytokine, right, is actually binding intramolecularly to the antibody and being guided towards the particular cell subsets of interest and biased in that way. Without going into super detail about the sort of synthesis and production of this molecule, we ultimately replaced the S4V6 antibody with an antibody called F10 that's against human IL-2 instead of mouse IL-2 and also is enhanced for its biasing properties. And specifically what we saw is that on CD8 positive T cells, this molecule was a little bit weaker than just native IL-2. But the important thing was when you went to CD4 and particularly regulatory T cells, you saw a 5 to 6 log, so a millionfold decrease in the activity of this molecule on Tregs compared to native IL-2. And this ultimately led to a very strong in vivo expansion bias in favor of effector cells over regulatory cells. So this looks at the ratio of CD8 positive to Treg cells. You can see whereas IL-2 does pretty much nothing in following 4 doses in a mouse, you can see that the F10 immunocytokine is bolstering that by a factor of 5 to 10. And same thing with the NK to Treg ratio. Ultimately, we wanted to make this into a therapy, and although the molecule is biased, it won't necessarily get to the site, right, of disease that we would want. So to this end, we actually were fortunate around this time that there was a paper published by Jeff Hubbell's lab, Juni Shihara specifically, who was a postdoc in the lab in Science Translational Medicine, showing that IL-2 conjugated to what's called a collagen binding domain, or CBD for short, actually leads to that molecule homing towards regions that are very high in collagen. And those in most people's body, areas of exposed collagen tend to be tumor tissue. And so based off of this, they had actually shown that IL-2 can be a lot more specific to the tumor, resulting in significantly inhibited tumor growth and extended overall survival in multiple mouse models of cancer. So we wanted to recapitulate this effect, but now with a biased IL-2, specifically the F10 immunocytokine fused to collagen binding domains. So we actually said we have some free C termini. So we actually took all 4 C termini in the antibody and fused them to the collagen binding domain. And what we ultimately found was that fusion to the collagen binding domain actually led to tumor localization and retention. So here we were doing intratumoral injections, so getting it where it needs to go from the start. But you can see with the F10 immunocytokine, it's escaping from the tumor environment over the course of 3 days, whereas the CBD is staying a lot more consistently in the tumor. And you can see that in terms of the inflection of the PK curve. But what was even more striking was when you look in the peripheral blood, whereas F10 is coming out, again, within a 24-hour period and then tapering off, there's no detectable levels of the collagen binding domain fused F10 immunocytokine throughout the duration of this experiment for 7 days. Ultimately, that resulted in significant inhibition of tumor growth and the absence of any toxicity in both a B16F10 mouse syngeneic model of melanoma, as well as a mouse syngeneic model of colorectal cancer, the CT26 model. And in combination therapies, when we treat with IL-2 to activate the immune response, and then also concurrently treat with immune checkpoint inhibitors to prevent the T cells from getting exhausted, we can see very strong synergy between the two. So if you have either one alone, it's this overlapping black curve and then dotted, blue curve and then dotted black line representing either the immunocytokine alone or the checkpoint inhibitors alone. You can see that there's extreme improvement in overall survival, this red dotted line, for the treatment with both the F10 immunocytokine with the CBD fusion, plus the anti-PD-1 antibody representing the immune checkpoint inhibition. So overall, this molecule shows a lot of promise as a cancer therapeutic. And then on the flip side, we also have this F5111 molecule that is biased towards regulatory T cells, right, in the opposite direction, that could be useful for autoimmune disorders. And you can see here that, again, whereas the molecule loses some level of potency on the regulatory T cells, it's completely inert on both the CD4 and CD8 positive effector T cells. Ultimately, this leads to a significant increase in the ratio, in this case, flipping the ratio from Treg to CD8 or Treg to NK. Typically, in most people's healthy human blood, you have like a 20-to-1 ratio of your CD8s to your Tregs, but we're actually flipping that completely to now be an 8-to-1 ratio of Tregs to CD8. So it's huge, massive expansion of the regulatory T cell compartment. That ultimately results in reduced disease severity in a model of ulcerative colitis in mice. Specifically, this is the dextran sodium sulfate model, where you actually deliver dextran sodium sulfate to the drinking water of these mice to induce colonic inflammation and ultimately a colitis-like disease. You can see that the weight loss in these mice is significantly mitigated in the purple curve for Fi111 immunocytokine delivery. And also the disease activity index goes down, and the overall morphology and histology of the colon is greatly improved in the Fi111 immunocytokine case compared to the significant damage you see when the mice are untreated or treated with saline or treated with a control immunocytokine that consists of IL-2 fused to an irrelevant antibody to extend the half-life without the bias. So we then looked in a model of type 1 diabetes, another autoimmune disorder where your body's own immune system is attacking your pancreatic islet cells. What we saw is that the immunocytokine prevents diabetes development. Indeed, when we looked in sort of a shorter term model on the order of 6 weeks, looking at anti-PD-1 or immune checkpoint inhibitor induced diabetes to accelerate and sort of homogenize the development of disease. You can see in the black curve with the control immunocytokine, as well as in the red curve with PBS, the mice very quickly become glucose intolerant, are no longer able to regulate glucose and develop diabetes. Whereas the purple curve actually shows this significant delay, and that 60% of the animals remain disease-free after 6 weeks. We asked if we went further in this experiment to the point where actually spontaneous disease develops in the healthy animals that were, or this is a NOD mouse model of diabetes, and the animals begin to develop disease spontaneously. We can see that our molecule actually protects for 6 months. This is looking 100 days, 80 days out from when they were first treated or when they were given their only treatment. And we've reshaped the immune environment so much, even 6 months out, still that same 60% of animals remains disease-free. So this is really exciting, and actually both of those molecules are patented, and we're actively developing those into potential clinical drugs. Okay, moving on to the second story, and this involves kind of the second arm of the lab, which is antibody-based design. Antibodies have a lot of capabilities, and one of their little known and little appreciated capabilities is their effects on trafficking and the way that molecules shuttle in and out of the cell. So a multi-specific antibody can actually be used as what's called a multi-paratopic antibody. And the way that that works is if you have a single antibody, because it has two arms and it's bivalent, it can bind to two different receptors on the surface. But that's it, right? It'll cross-link them as a terminal dimer. If you come up with another antibody that binds to the same receptor, right, that navy blue receptor, but actually at a different site, you can actually cross-link together all of those receptors, right, making these big antibody receptor clusters of potentially infinite length, right, around the surface. And that's important because that can manipulate the overall trafficking of that receptor. So receptors, right, will endocytose at some rate. They'll then recycle back to the surface, and they have a synthetic rate. And then when they're internalized, they can also be shunted towards degradation pathways. And what's interesting is that when when multiple antibodies in combination that bind to the same epitope are added and cluster the surface, you see a significant increase in endocytosis, significant decrease in receptor recycling, leading to massive downregulation from the surface and ultimately degradation of those receptors. You can actually achieve this by looking at creating a multi-specific antibody, right, that actually does the same thing in one molecule. So here you can see there's one red arm and one teal arm, and now you can actually use a single molecule to achieve this multi-paratopic cross-linking effect. So what's ultimately useful? This can greatly decrease the availability of the molecule for signaling. So one arm can be basically doing what antibodies normally do, which is neutralize by sort of coming in between an interaction between a ligand and a receptor. And then the other arm, downregulation, so that by acting on both the ligand side as well as the receptor side, you can synergistically reduce availability of your molecule for activation. And this could be useful potentially in the context of immune checkpoint receptors. And the reason is, or immune checkpoint proteins. And the reason is that when T cells get activated, right, there's three signals that they see. Signal 1, the peptide MHC, signal 2, co-stimulation, and then signal 3 is cytokine secretion. But that leads to upregulation of various exhaustion markers, such as PD-1 on their surface, which can be activated by tumor cells overexpressing PD-L1 ligand, and inducing that T cell to become exhausted. As a result, people developed, of course, immune checkpoint inhibitors, which are monoclonal antibodies that block PD-1 binding, and that results in reactivation of the T cell, such that it can ultimately be able to be reinvigorated to then go and attack cancer cells or infectious diseases, or cells infected with an infectious disease. And ultimately, that same thing can be achieved even better using a multi-paratopic antibody, right, which not only will come in between the PD-1 and PD-L1 interaction, but also directly downregulate PD-L1. Okay, so if we want to discover a multi-paratopic antibody, we want something that does not bind to the normal PD-1, PD-L1 binding site. And specifically, we want to discover a multi-paratopic antibody against PD-L1 that has two arms or more that would bind to unique epitopes. So to this end, we actually did a discovery campaign, two discovery campaigns, one using a generic scFv library, and then using the yeast display platform. We selected for things that would bind against the PD-L1 extracellular domain. And then we actually also had the opportunity to immunize nurse sharks in collaboration with Helen Dooley's lab at the University of Maryland, Baltimore. And with that collaboration, we were able to discover two different nanomolar affinity, what are called VNARs, or single domain antibodies from sharks, and ultimately use those to create multiparatopic antibodies. And what we found was, whereas the PD-L1 PD-1 interaction is inhibited by the current commercially approved immune checkpoint inhibitor antibody known as atezolizumab, that binds to the PD-1 binding site of PD-L1. And whereas the D12 antibody, which was discovered from the scFv library, is actually competitive with atezolizumab, we found two VNARs called A9 and B8 that were actually non-competitive with atezolizumab and non-competitive with each other. So we could actually make as much as triparatopic antibodies. So I had a very talented graduate student, Seth Ludwig, who actually made this whole panel of biparatopic antibodies and showed that they downregulated IL-2 or downregulated PD-L1 to varying degrees. Specifically, this BS15 was sort of our winner. And off of that, we actually developed another panel of triparatopic antibodies, including 3 unique epitopes. And again, there were varying levels of downregulation for each molecule here. And the winner was this molecule that we called TS1521. We then demonstrated that the TS1521 actually was effectively downregulating in many different contexts. And specifically, this microscopy image shows the co-localization of the PD-L1 shown in green with endosomes and lysosomes. What you can see is that there's increased localization of TS1521 compared to just the atezolizumab parent antibody in both endosomes and lysosomes. And I don't know if this video is going to play, probably not. But you can actually see those punctate spots shown in green on the TS1521 that are representative of the surface clustering of the PD-L1 in the case of TS1521 administration compared to atezolizumab administration. Ultimately, we wanted to see if this would actually lead to decreased bioavailability of PD-L1 in the tumor microenvironment. And to this end, we had a radio-labeled peptide. This was in collaboration with Sridhar Nagada's lab at the Johns Hopkins Hospital. And what we showed was that the radio-labeled peptide, which was competitive with PD-1, could actually be decreased by pre-treatment with TS1521, reducing the number of available spots for that radio-labeled peptide to interact. So what we did was we injected just into the tail vein, so systemic injection of TS1521, and then we monitored the ability of that TS1521 to actually block the bioavailability of PD-L1 in the tumor. So you can see in the control animal, there's extreme amounts of the radio-labeled peptide in the tumor microenvironment. There's significantly reduced amount of PD-L1 shown on the right when you pre-treat with TS1521. And if you quantify that, you get a very significant drop, about tenfold in terms of your control mouse versus your TS1521. And after 96 hours, there's a little bit of a rebound, but you still have a very significant drop in your overall bioavailability. And so there's clearly a sustained reduction in the availability of that molecule. And then finally, we wanted to show that by blocking the availability of the PD-L1 for signaling and for stimulating the PD-1 binding partner, that we could actually reactivate and reawaken T cells. So to this end, we actually used T cells from patients with hepatitis C. And this particular cohort of patients was known to have very high levels of PD-L1 on their T cells. So this would be a good test case for our molecule. And we treated those patient samples with CMV peptides. These were patients who were CMV positive, so that we could stimulate signal 1 and ultimately get immune activation. We did that in the absence or presence of our various antibodies, and then performed ELISpot to look at interferon gamma levels. And Ultimately, as using interferon gamma as a readout for T cell activation, we saw that whereas atezolizumab did lead to some modest increase in the interferon gamma secretion, the TS1521 shown in purple led to a significant increase compared to both the atezolizumab and the control in terms of overall levels of interferon gamma secretion. That was very exciting. Okay, finally, in the couple minutes that I have left, I'm just going to high-level touch on the third project that I mentioned, and this goes to the third arm of our lab, which is on the platform development side. And this is developing a system or infrastructure for discovering antibodies that bind to membrane proteins. So membrane proteins really pack a punch. They make up only 23% of the human genome, but actually the drug development landscape, actually 60% of drugs are against membrane proteins. And the reason for that, of course, is that membrane proteins are critical for sensing information on the outside of the world and bringing that inside the cell. And so these critical molecules, right, could definitely use some antibodies or other types of targeted drugs against them. But unfortunately, in order to discover antibodies, as we did in the shark case, you have to immunize an animal. And the question is, with a membrane protein, how do you immunize an animal with a membrane protein if the membrane protein can't be folded, right, or expressed outside of the body? In particular, there are many purification well-documented purification challenges with particularly multi-pass transmembrane proteins like GPCRs. And then when you actually do express them, they may or may not be in their native orientation with their native glycosylation patterns. And that also can create kind of a moving target that's going to make any sort of drug development much less efficient. So we figured, why not actually bring the library to the cells themselves expressing the protein rather than trying to purify the protein from the cells. So that's exactly what we did. And we combined mammalian cells expressing our target protein with yeast cells expressing an scFv library for high throughput antibody discovery. And without going into too much detail, we're not the first ones to think about this idea. Previously, there was an idea known as biopanning, where you would just throw the yeast library on top of the mammalian cells, let it settle, and see what bound, right, after several different washes. And that was very laborious to do in terms of both the technique and then also just the measurement and quantitation. Whereas for biofloating, where everything was done in solution, you could actually quantify that by flow cytometry, parallelize things a lot better, and get better sensitivity. So the ultimate goal was to use that, right, to be able to select out of a naive library anything that would specifically bind to your target cell expressing your protein of interest. And so using very iterative rounds of max and fax that we optimized for various parameters, we were ultimately able to come up with a selection strategy to discover antibodies from a naive library. And that involved basically selecting for things that were specifically binding to the protein on the surface, but not binding to some other random surface protein, as was seen in the target null versus target expressing cells. And ultimately, we settled on the use of one negative pre-clear round where we label the target null cells and pull out everything that binds to them and get rid of them. We then actually add the target null cells as sort of a decoy, and then also add target expressing cells so that only the best binders, right, the ones that are very specific to our target, will be able to bind. We then discard our non-specific binders, and ultimately we can pull out the binders that are specific for our target cells. We iterated this process for a variety of different GPCRs. We started with the CXCR2 GPCR, which is a chemokine receptor for the IL-8 chemokine. What you can see is over the rounds, you get increased ratio of binding to target expressing versus target null cells, shown in blue and red respectively. And that ultimately led to increased specificity towards our target cells. And it turns out that actually we found about 27% of the binders that we pulled out of this library were specific to our target. And in fact, we actually found things that bound at the at epitopes other than the N terminus, the long and flexible N terminus that IL-8 normally engages with. So that was very exciting to sort of observe. And then also we found things that were non-competitive with that N terminus for the first time using this unique platform. We then tested our platform on 3 different GPCRs, GLPR1R, GCGR, and CXCR4. And in all 3 cases, we were able to get specific binders in the range of 60 to 100%. And overall, if you add up all our campaigns together, you can see about 72% of the binders that we got out of yeast were specific to our target, and 80% of those wound up expressing and holding up recombinantly. So unlike a lot of other yeast display platforms, what you see is what you get, right? And we were really able to pull out from naive libraries bona fide binders against our target. Okay, so wrapping this up to keep within time, I talked about 3 stories today. One was about cytokine antibody fusion proteins, or immunocytokines as they're called, that allow for selective and biased delivery of IL-2. The multi-paratopic antibodies story, right, talked about targeting PD-L1 to induce rapid and robust targeted downregulation. And then finally, suspension-based cell-cell interaction platform called biofloating that we developed allowed for high-throughput antibody discovery. With that, I want to thank the amazing people who actually did all of this work. A wonderful research faculty in my lab, Jacob Tomala, was very involved with immunocytokines, as were Alyssa, Joe, and Derek and Aaliyah. Pao and Seth did a lot of the work on the multi-paratopic antibody, as well as Ben. And then PJ and Justin have been the ringleaders on biofloating. Thanks so much to collaborators, particularly my postdoctoral mentor, Chris Garcia, where we solved the crystal structure of the anti-IL-2 antibodies, and then additional collaborators from around the world, as well as from here at Johns Hopkins. Thanks to all of you for your attention, and excited to engage in the Q&A.
All right, excellent. So thank you very much for your presentation. We're going to go ahead and get into the live Q&A portion now. So if you do have any questions that you haven't sent in, please do so now. You can submit them in the Ask a Question box at the bottom of your screen. So let's go ahead and get started. First one I see here is, what are major challenges you face in translating molecular therapeutics from lab to clinic?
Great question. And actually, bioprocessing manufacturing, right, is a big hurdle that you need to get past. For a lot of these molecules, they look fun, but they're not always a pleasure to make. And so, really finding and optimizing the formulation, and then finding ways to purify them, right? If you're someone who can get what you want out of lots of things that you don't want, you're in very good shape. And that will make you a very rich person. And so in a lot of cases, it's having the right tags, the right combination of tags, to make sure that the molecules can be pulled out of a solution, and that the molecules that you get out of that solution are exactly what you want and not things that you don't want, and that they're in a very pure state. Another big hurdle, right, can be actually getting the support behind the molecule, because obviously to do all of these different tests, scale up manufacturing, toxicity, immunogenicity, develop all these methods requires money. And whereas pharmaceutical companies, that's their bread and butter, for academics, right, our funding source mainly comes from government grants, and also foundation grants, and other sort of funding mechanisms that are meant for more basic and innovative research rather than just kind of plug and chug away at making a drug, because that's generally more suited for pharma. So our lab and others are really looking at ways to kind of integrate with pharmaceutical companies and work in concert and in coordination, so that we can each play to our strengths and ultimately find the right balance and also the financial support to sort of move things forward.
All right, thank you very much.
Yeah, great question. Thank you.
Let's see here. Which types of proteins are most amenable to target discovery using suspension-based cell-cell interaction platforms?
Yeah, what's really nice is pretty much any membrane protein that we've tried, we've gotten hits out of and we've gotten results out of. Of course, if you have a single pass transmembrane protein, that does simplify things in terms of the expression and in many cases, the discovery process. But we've tried, a variety of different targets, GPCRs, ion channels, glucose transporters, all kinds of different metabolic, sort of proteins and molecules. And we've had a lot of success so far. So I think really what you need is just a very clean cell line, right, that expresses a lot of your target. You have to have at least about a few 100,000 copies of your target per cell in order to have a robust selection procedure. And then if you have that, and then you have a target null cell line that's completely knocked out, that particular protein, then you're in business. And we've had a lot of success with many different campaigns.
All right, excellent. Looks like we got time for one or so more. So let's go with, are there additional cytokines that are amendable to immunotherapeutic development?
It's a great question. So there's actually many cytokines. There's 40 interleukins, and then 30 interferons. So there's many different, and then there's chemokines, growth factors, all kinds of different things that we could discover, binders for. So we think about immunocytokines, right, fusing antibodies to cytokines to create these pharmacologically favorable and translatable molecules that could go in a lot of different directions. So our lab has actually been exploring other cytokines as well as other growth factors, chemokines, as potential partners for fusion to an antibody and ultimately bias of that molecule through intramolecular interactions and using those as potential therapies. So I think there's a lot of opportunities, some of which we're pursuing and some of which I hope other people pursue, kind of inspired by some of the work that we've done.
All right, great. Well, thank you again, Jamie, for your time and your research. Before we go, I'd like to also thank the audience for joining us today and their questions. Any questions we didn't have time for today or those that get submitted during the on-demand period will be addressed by the speaker via the contact information you used when you registered. The webcast will be on demand for a year, so you have until April 2nd, 2026 to watch it. LabRoots will alert you via email when it's available for replay, and we do encourage you to share that email with any colleagues who might have missed today's live event. So until next time, thank you very much and goodbye.